Hi guys, welcome to my presentation on DNA organization, replication, and repair. This presentation goes over chapter 35 of Harper's Illustrated Biochemistry. So DNA contains the genetic information of our cells. It's basically an instruction manual that our cells need in order to create proteins and perform basic functions. It's very important. And it's also very long. If it was stretched out, it would be about 6.5 feet long. So it can't just be floating around in our cells because lots of things would go wrong. So because it's so long and there's so much of it, it needs to be organized. And thankfully, our bodies have a very complex and very nice way of organizing DNA. So the way DNA is organized is by wrapping around histone proteins. DNA is inherently negatively charged because of the phosphate groups in it, and histones have a positive charge. So these DNA molecules will naturally associate with the histones and wrap around them. DNA wrapped around histones is called nucleosomes. Together, they form chromatin, and once chromatin is packed together, this is called chromosomes. We have two types of chromatin in our bodies. There's euchromatin, which is more loosely packed, and it's more easy to access by transcription factors, so it's more active in transcription. We also have another type called heterochromatin. It is packed more tightly and close together and harder to access, so it's usually inactive. And as I mentioned earlier, chromatin makes up chromosomes by condensing together further. They are basically highly condensed DNA strands. There are a few important locations on our chromosomes. One important location is a centromere, which is in the middle of the chromosome. They hold sister chromatids, which are identical chromosomes together, and during cell division, they're very important to ensure proper separation so that each daughter cell gets the proper amount of sister chromatids. There's also another important place called telomeres. They are the protective ends on our chromosomes. They contain repeating sequences, and each replication cycle, they shorten over time. And so towards the end of our lifespans, telomeres are very short, and it leads to the stopping of replication, cell death, and a halt in behavior. So where is DNA? We have so much of it. Most of it is located in the nucleus of our cells. That is the main area our DNA is. But we also have a little bit of DNA in our mitochondria. Unlike the linear DNA molecules, it's circular. An interesting thing about mitochondrial DNA is that it's only inherited from our moms. During fertilization, mitochondria in the sperm are usually destroyed. And so all the genetic information we have in our mitochondria actually comes from our mother's mitochondria and her mother's and so on. No mitochondrial DNA is found from the father's DNA. It's also replicated independently of nuclear DNA. So cell division is something that's very important in maintaining proper bodily functions. But before cell division, DNA needs to be copied so that it can be transferred to new daughter cells. Replication is semi-conservative, which means each new DNA molecule has one old strand of DNA and one new strand. So each strand of DNA that we have is actually a template for a new strand. Replication is a little bit of a complicated process, and there's lots of different factors involved in replication. But it all starts out with helicase. The DNA becomes straightened and it's unwound by an enzyme called helicase. After the two strands are separated, something called DNA primase comes in and adds a short RNA primer in order to begin the replication of DNA. After this, DNA polymerase comes in and begins replicating the DNA using complementary base pairs in the 5' to 3' direction only. Because there's two different strands in our DNA, this means that both strands cannot be synthesized continuously. There's a leading strand, which is synthesized continuously in the 5' to 3' direction, but there's also a lagging strand, which is replicated in short fragments. These fragments are called Okazaki fragments. And because the DNA is synthesized in short fragments, there needs to be something to bring these fragments together into a continuous strand. And that's done by DNA ligase, which also serves other functions important in DNA repair. Replication is very important, and so it needs to be accurate. Because of this, there are specific base pairs that will only bind to other specific base pairs. Adenine binds to thymine, and cytosine bonds to guanine, as you can see in the picture. These base pairs will only bind to each other And if there's a mismatch, like let's say if cytosine binds to thymine, there are repair systems in place in order to correct that. So let's move on to the concept of DNA repair. Sometimes DNA repair is needed. Damage to DNA could cause cells to stop dividing, to die, or lead to critical errors in protein production, which we really don't want to happen. If the wrong proteins are synthesized in our bodies because of something like incorrect replication or a replacement of base pairs or breaks in our DNA, it could lead to major cascading problems in our bodies. So we really want to avoid this. And because of this, there's lots of complex systems that our bodies have in place to fix these errors. So now let's go over the different types of DNA repair. The first type of DNA repair happens the earliest in this process, during DNA replication. Now, DNA is synthesized in the 5' to 3' direction, but if an incorrect base pair is added, there's a system that our cells have in place called exonuclease activity. Exonuclease activity fixes errors during replication, and they can move backwards 3' to 5' to remove the incorrect nucleotide. It's a form of natural proofreading that our bodies have in order to minimize the errors during replication. Now, if exonuclease activity doesn't fix an error during replication, there's things that can be done after replication to correct incorrect base pairing or other types of things like insertions, deletions, or breaks in DNA. The first system in place is called the mismatch repair system, and it can fix incorrect base pairing after replication. The mismatch is first recognized and then cut out. Then DNA polymerase comes back in to put in the correct base pair, and then ligase comes and seals the nick. If there's any damage to our bases, there's something called base excision repair. First, the damage is recognized, the damaged base pair or changed base pair is cut out. Once again, DNA polymerase comes and fills the gap, and ligase seals it. For larger sections of damaged DNA, we have something called nucleotide excision repair. First, the damage is recognized, and a larger area of DNA is cut out. DNA polymerase fills in the missing DNA section using the complementary strand as a template, and then DNA ligase comes in to seal the nick that was caused. For more serious double-stranded DNA damage, there's something called homologous recombination. However, it can only happen when the other sister chromatid is available to use. Whenever there's a double-stranded break, DNA polymerase can't come in and just seal the gap because the template strand is also damaged. Because of that, there has to be other methods. Instead of using the template strand as a template, we have to use the other homologous chromosome as a template. Because this isn't always available, we do have a last resort for serious double-stranded breaks, but we really don't want to use it unless we have to because it could lead to major problems. This last resort is called non-homologous end joining, and it directly joins the broken DNA ends together. As you can see, it's not the best option because it can lead to mutations and synthesis of incorrect proteins later down the line because we don't have the right base pairs anymore. We can only hope that once these ends that aren't broken join together, it can produce the same or similar proteins. The best case scenario whenever non-homologous end joining needs to be used is if the damage happens in heterochromatin, which is inactive during transcription. Hopefully, from this lesson, you learned that DNA is very important and there's very complex mechanisms in order to store DNA, replicate it, and to repair damage. DNA has information necessary for organisms to develop, survive, and reproduce, and the organization, replication of repair protects our genetic information. Problems in these processes can lead to mutation and disease. Well, I guess you guys can see the answers already. It wasn't supposed to do that. But the first question is, what is tightly packed inactive DNA called? And the answer to that is heterochromatin. Euchromatin is the active form. Histones is what DNA binds around, and nucleosomes are what it's called whenever DNA associates with histones. The second question, What is a feature of mitochondrial DNA? The correct answer is it is inherited from the mother. Remember that our mitochondrial DNA only makes up about 1% of our DNA. It's not inherited from the father because mitochondria in sperm is destroyed during fertilization, and it is not replicated with nuclear DNA, it's replicated independently of nuclear DNA. How is the leading strand synthesized during DNA replication? The correct answer is continuously in the 5' to 3' direction. Remember that DNA is synthesized in the 5' to 3' direction, and it's not discontinuously. The lagging strand is produced discontinuously. We also need DNA polymerase in all of our replication, in both the leading and lagging strand. Next question, what is the purpose of DNA ligase? The correct answer is it seals, breaks, or nicks in the DNA backbone. DNA helicase unwinds the DNA double helix. Primase adds RNA primers. And while DNA ligase does serve a purpose in DNA repair, it does not remove damaged bases. The final question is, what type of DNA repair would be used to correct a single base pair mismatch after replication? The correct answer is the mismatch repair system. Base excision repair does happen after replication, but it's only for a small damaged or mismatched nucleotide. The mismatch repair system is a little bit different. Non-homologous end joining, as I said earlier, is the last resort, and we really don't want to have to do this for just a single base pair mismatch. An exonuclease activity does correct a single base pair mismatch, however, it happens during replication. These are my references. I hope you guys enjoyed the presentation and learned something new today. Thank you.